Next speaker would be Dr. Andreas Kurtz. Dr. Andreas Kurtz has been the director of the German Human Embryonic Stem Cell Research Authority and heads the European Embryonic Stem Cell Registry, as well as the data management at the European Bank induces pluripotent stem cells. He has developed the tool CellFinder to support the cell-based application and facilitate long-term cell-related data storage. As a faculty of the Berlin Center for Regenerative Therapies, he is carrying out efforts for the translation of stem cell research into clinical application. In addition, he is a faculty of the Berlin School of Regenerative Therapies, an international graduate school fusing biology, clinical, and engineering tracks. Professor Kurtz is also faculty at Seoul National University with a focus on biotechnology, bioinformatics, and the application of stem cells for regenerative therapies. He is going to speak about the human pluripotent stem cell registries and the CellFinder from registry to research tool. Dr. Kurtz, Andreas, please go.
Okay, can you see my presentation?
Okay, now you can see you well.
Okay.
Please go ahead.
Okay, thank you very much, Nancy, and thank you for all people who were helping organizing this event, which I find very interesting and exciting. So I want to present my two systems. One is called HG2, it's a registry of stem cells, and the other is called CellFinder. And both are used to basically collect data on cells. And the reason why we do that as a translational institute in Berlin is to have more information on cells available for clinical trials or tissue modeling in vitro. And this is necessary to compare results of clinical trials or compare cells with regards to their capacity to do certain things. So if you look at this tree on the left side here, you can see that on the bottom there is a cell from which all other cell types in the body can arise. These are called pluripotent stem cells, and the other cells are either other stem cells or terminally differentiated cells. All these cells can be used for several applications, including regenerative medicine, tissue modeling, which can again be used, or disease modeling, which can again be used for drug testing or toxicity testing of new drugs. The problem is, in the moment, that we have a lot of data available on these cells, but we have no way to collect these data in a common and comprehensive platform. So one more slide I want to show on this background, and this is how these cells can be used. So in our institute, we define regenerative medicine in kind of two ways. One is to directly use cells in the patient, and the other way is to stimulate cells which are in vitro. It's called endogenous support of regeneration, which are already present in the body to repair better than they would normally do. But for both aspects, we need to understand better what cells are actually doing and what they are. So in the simplified pattern here for kidney, we can use cells for direct regeneration. We can use these cells for modeling and then drug testing, but for both of them, we need some kind of application to knowledgeable define what cells we are using and also to get feedback from the application with these cells. So as I mentioned here, we need these in silico systems for modeling of cell interaction of cell type and cell identity definitions, for standardization, and for cell comparison. So having said that, what kind of data are there for cells? And there are a multitude of data. It's an amazingly growing body of information we have. This includes sequencing data, protein data. They come in different shapes and forms, as tables, as spots, as graphs, or images. So we all know these anatomical images from cells. also other types of images like pictures from the microscope taken from an in vitro, in this case pluripotent stem cell, or other information which is available. And the challenge here is, if you want to comprehend this information, to put them in a database. This is basically the database which we have here. All the cell data are collected from different sources. So one is the user and the applicant. So the user and applicant gives us the data. We have a certain cell which performs in a certain way, and here is the information we collect. We need the data tools in the system which enable the user to analyze the data and compare cells. And we have a close contact to cell banks. And now about in the next 5 years, I expect to be 20,000 pluripotent stem cells being banked around the world. And the information of these the cells banked need to be compared at a global level. So we don't need to go to 10 different banks and different nations to find the cell you're looking for. So this is the task of a registry. So what cells are available, what data are available for each of these cells, and where can I find the safe and store my data on the different cell types. Now I'm going to talk a little bit about the two systems more specifically. The one is HG2. It's a European funded project on pluripotent stem cells, which has an aim to collect data on all these pluripotent stem cells that are available, and these are the pluripotent stem cells here on the top of this tree. From these cells, all the other cells can be derived, and to get information from all the other cells, we have a different tool which is called CellFinder. So if you don't have a registry, this kind of embryonic or pluripotent stem cell registry, what would be the risk to the field? Of course, we don't have a collection of peer-reviewed and new cell lines. That's the first risk, that it would not be there, and we would probably have a widespread use of cells without appropriate ethical and scientific validation. Finally, we would have a widespread use of cross-contaminated cell lines. So that's a big problem. In a recent study, it was shown that 30% of the cells being used in the laboratory are cross-contaminated actually with other cell types. And we would have, of course, a difficulty to locate certain cells, which then might be a resultant application of research and waste of money, basically. The pluripotent stem cell registries are very simple selection process for the cells that will be registers. So these are the 3 points. One is these cells must be available for public domain research. There must be evidence of informed consent for the donation of these tissues used for making these cells, and there must be certain characteristics fulfilled. In the case of pluripotent stem cells, it must be clear that these are pluripotent stem cells. So I just want to now, because of time limits, I would like to show you how we do that. So to have all these different data types collected and organize the user in ontology, which is a structured hierarchy of logically connected classes. I don't want to go over that example, but basically you can see that the embryonic stem cell line, which you might have here, is a subclass of other ontology types like human cell line, inner cell mass, embryonic structure, and so on. So we use for this newly developed ontology already existing ontologies and just combine them to a big ontology which includes all cell types which we know. So this is then put in a database which has several parts: ontology, of course, a knowledge base, which are the actual data experiments, definition, references, and relations. So the database also includes, for example, geonames, so we can say which cell comes from which country, to which disease is a certain cell associated if it comes from a certain patient. We have the ethical background information for these cells. We have information on the derivation of these cells, genotype, karyotype, and so on. So this is HG2. So HG2 collects the data of all these pluripotent stem cells. What about the other cells which are down here? This is the pluripotent cell, and the differentiate in certain all other cell types of the body, and we need the information of these cells as well. So this is put in CellFinder, which comprehends all the data for cells in vivo as well as in vitro. And to visualize these, or to browse all these data, we have two tools. One is called Body Browser, and one is called Developmental Tree. This is a body browser where you can basically click on the human body on a certain tissue where the cell of interest should be or might be located. And then you get here all the information on the different cell types present in the certain tissue. The developmental tree shows you, on the other hand, the derivation of that cell from a stem cell, for example. How can we get from a stem cell to a certain differentiated cell types, or reversely, how is this differentiated cell being developed? So we can visualize that, and then we get a table with all the information for certain cell types associated to that. In the case of a kidney, for example, this is kidney development. Maybe here it's better seen, consists of two cell types, which then form the whole kidney. This is a mesenchyme, and this is a uretary bud, two cell types. And we can now go to CellFinder and say, what kind of cell types, what information do we have on these cell types? And we see in the body browser the kidney, substructures of the kidney down to the single cell of the kidney, and we can ask the question, so what kind of information do we have? How does that cell compare to the cell I have in my petri dish, and how can it be used for clinical application? This is just higher resolution pictures of this body browser images of a kidney structure, which is associated with real images and information which is available in the database. We also have included text mining results. This is a snippet of these text mining data which are annotated manually. And to conclude this, I would just like to mention four things. We offer, these two systems offer a way to store data on cells for a long time in an organized manner. These data are validated and curated. We have data tools for the analysis and searching of these data in cells and comparing cells based on these data. And of course, that includes a wide variety of data. So I hope I could give you an impression of what we are doing and what these two systems can do and might be useful. So thank you very much. Any questions?
Okay, so please change the video screen. So this is all the screen. So hello. Please stop sharing your button. Okay. Okay, thank you. Okay. Thank you for your presentation. So, is there any question from the? Also, I have learned from Professor Suik Chang's lab participants now in Cheongju Chungbuk National University. Do you have any question to Dr. Andreas Kurtz?
Yeah. Hello.
Okay, please go ahead.
Yeah, actually, I wanted to know whether this body browser program can give the information how to program a embryonic stem cell to a particular cell type. Hello.
Yeah, no, I understand your question. So you want to have the specific protocols for differentiating a certain pluripotent cell and other cell type. We don't have that in the moment, and but we have planned to include these data, which needs basically to read all the publications available on this differentiation protocols and include them in the moment manually into our system, which takes some time. But yes, this is an important question, and there's high interest on that. We will do that in the frame of the pluripotent stem cell registry, because we think the users who want to differentiate pluripotent cells will first go there. But the backbone for both CellFinder and the registry is the same. So yeah, yes, we will have that. We don't have it in the moment.
Okay, maybe in the future.
Okay, okay, thank you, Dr. Andreas Kurtz. So actually, what time, or what is your local time? So it's early morning?
Yeah, it's early morning. It's going to 6 a.m.
6 a.m. Actually, it's Berlin or London? Where are you now?
Yeah, I'm in Berlin now.
Okay, thank you. Thank you again for your early time participation. Okay, thank you.
Thank you.